We've almost made it through the chemistry part of our lectures. We have so far discussed carbohydrates, we've discussed fats, lipids, which included triglycerides and phospholipids and steroids. We've discussed proteins. We know that proteins are polymers built out of monomers that are called amino acids, and that they are amazing machines. So what is there to talk about? Well, nucleic acids. This is our fourth biomolecule that we needed to discuss. And nucleic acids are built they are also polymers, and they are built out of the monomers that are called nucleotides. All right. So to me, that was a little bit confusing because proteins are the polymers built out of amino acids. But when it comes to these, nucleic acids is the polymer and the monomer is nucleotides. So find your way of remembering that. Now, each nucleotide has got a lot of things that it shares with other nucleotides. Every nucleotide has got a 5-carbon sugar. We remember glucose was a 6-carbon sugar, right? Nucleotides have a 5-carbon sugar. We'll talk about those. They've got a phosphate group. Oh, that's kind of like phospholipids, right? And then attached to it is something called a nitrogenous base, The two basic nucleic acids that you should know are DNA. And DNA is the stuff that makes up our chromosomes, mostly. And then the other one is called RNA, also plays a role in what is known as genetics, but a more subtle role. Okay. This is what a little teeny tiny fragment of DNA would look like. But I like this. Okay, so DNA. DNA is in the structure of a double helix. A helix is like a spring kind of shape. It's made out of 2 strands. So here's strand number 1, and here's strand number 2. And they are held in the center like this. By a bond. Can you remember what kind of bond? You're going to need to know it for exam 1. Check your notes. Okay, it's a hydrogen bond. It's held together in the center by hydrogen bond between that guy and that guy, between that guy and that guy, between that guy and that guy, held together by hydrogen bonds. All right. And this is I told you, a teeny tiny snippet of a molecule of DNA, because a molecule of DNA has got like 50 million base pairs, and this is 1 base pair. So 1, 2, 3, 4, 5. What is that? That's maybe 20 base pairs. 20 base pairs versus 50 million. Okay, this is a tiny bit of DNA. RNA it's shaped a lot like a single strand of DNA. It's not in the form of a double helix. Now, we're going to be talking really a lot about DNA and RNA and proteins. As a matter of fact, lab number 3 is all about DNA and RNA and how it is used so that we can build proteins without any mutations. All righty? So this is what one nucleotide looks like. So here's a nucleotide. Remember I said a nucleotide was the monomer that nucleic acids are built out of. So it's made out of a 5-carbon sugar, a phosphate group, and then hanging off to the side is a nitrogenous base. And for DNA, there are 4 different bases that could be used. In DNA, the sugar is called deoxyribose. That's where the D in DNA comes from. DNA is the abbreviation for deoxyribonucleic acid. So DNA, the N in DNA, deoxyribonucleic acid, DNA. Okay, we're going to talk more about that, right? So don't think they're unimportant because I just barely talked about them now. It's because we've got whole lectures on them in the future. In the meantime, let's go over a couple of sample questions. Let's see how ready you are. An atom with a negative charge, it has more protons than electrons. Does it have more electrons than it has protons? Does it have more neutrons than it has protons, or does it have more neutrons than it protons? That's the same thing twice. Hopefully my exams don't have typos like that. Alrighty, so what is it? Atoms with a negative charge. What are the negatively charged subatomic particles? Electrons. Electrons have a negative charge. What are the positively charged ones? Protons, right? If you've got the same number of electrons and protons, you have no charge. If you've got a negative charge, you've got too many electrons. So B. The answer is B. Good job. Which of the following is an example of a polysaccharide? Polysaccharides are things like glycogen or other starches, and so starch, right? Glucose, maltose, and fructose. Well, glucose and fructose are monosaccharides, all right? Good. What are saturated fats saturated with? Do you remember? Saturated fats all have got single covalent bonds, no double covalent bonds. Hydrogen, right? What are the major lipids of plasma membranes? Are plasma membranes made out of steroids? No, but we'll talk about hormones. Are they made out of fatty acids? Fatty acids, they are one of the building blocks of a triglyceride. Are they made out of triglycerides? No. Triglycerides, that's a storage form. There's that guy. Phospholipids. Phospholipids, they're the amphipathic molecules that make up plasma membranes. And remember, their hydrophilic heads are on the inside of the cell, and the outside of the cell, and their legs are pointing towards each other in the middle of the phospholipid bilayer. Now you might be saying, Tidal, you have said that at least 4 times during these lectures. That's because it's on the exam. Make a note of it, okay? Isotopes of atoms differ in their number of, let's see. They don't have, atomic nuclei, atoms have 1 nucleus, okay? The number of protons, no. Every element, if it's of that element, it's got the same number of protons. Ions, they differ in their number of electrons. Isotopes differ in their number of neutrons, all right? Okay, that's an example of the kind of questions that you might see. In the meantime, we will be moving on to the structure of cells in our next lectures. I'll see you then.